the life process of microbes helps it so that we can understand all of life. Also, microbes are incredibly beneficial. We make our antibiotics. They ferment our, otherwise without microbes we wouldn't have beer. And they're now doing this human microbiome. So they're actually studying what are all the different microbes within you. So there were some weird numbers like 700 trillion microbes in a human body. And so it's like, you're just kind of a placeholder for all these microorganisms. So how do we figure this out? What happened? And really the father or the pioneer in microbiology was Rudolph. So he was this lens maker, and he invented microscopes. So he would make these simple little one-lens microscopes, and he was able to look at pond water and go, there's things swimming in my water. This is also the time when science would not allow you were allowed to have sex, to have a kid. Any other secretions from a male was not allowed. And so he had to make up a lie, going, oh, I got this from my wife after the fact, and it was the first time that anyone saw sperm, or knew what sperm was. And it was all because he was being able to put them on a microscope and start describing these things. So he made very simple microscopes. He would examine water, so he could see animals, fungi, algae, little single-celled protozoas. And so these organisms were called microorganisms because they're small. You need a microscope to see them. So this is what a basic one looks like, very, very simple. He would often make a new microscope for every single specimen, so he'd just keep them, go back and look at them from time to time. But he didn't really like to share. He didn't tell anyone how to make them. He didn't even let most people or anybody look through them. His best ones, he wouldn't share. He wouldn't let, I guess apparently he even had like the czar come and visit him. He wouldn't let him look through it. And so because he was so secretive about his stuff, it took us almost 100 years to get microscopes of his quality after he died. So, looking through a microscope, crude microscopes, what did you see? So here some spiral bacteria, here we have some algae, maybe some protozoa. Okay, so just kind of like, what you can start seeing. And one of the things you also should start taking note of, it got cut off here, but 50 micrometers. So, what is a micrometer? Anyone know? What's a meter? Yeah. 10 to the negative 6 meters.
Zillion to the meter.
Hmm?
Zillion to the meter.
There you go. So, it depends on how you think it. I always do negative 6. It's easier math-wise. But if you think about that, that's a lot of zeros. We're shrinking these things down. So, everything we do in here is going to be in that microscopic scale. Okay, so, microbes. What are microbes? Generally, they're smaller than the eye can see. Some of them can be really big, and if you squint just right, you can pretend you saw it. But generally, you need a microscope. They get classified based on their taxonomic structure, and so they get grouped into similar organisms. The types that we're going to especially be covering in this class, and we have prokaryotes, which are bacteria and archaea. There are fungi, protozoa, algae, viruses, and then small multicellular animals, like parasitic worms. Some things we'll look at. Okay. So here is 2 cheek cells. And you can see the nucleus size of this cheek cell. So, eukaryotic human cheek cell versus a prokaryotic bacteria cell. So look at the size of these compared to the size of those. It starts to kind of give you a little bit of an idea. So, bacteria and archaea, prokaryotes. Prokaryotic means that they lack a nucleus. They're unicellular, means they have a single cell. All prokaryotes are single cellular. They are much, much smaller than eukaryotes, as prokaryotic organisms can be found anywhere there's enough moisture. And they reproduce asexually. So they just divide, make new ones. Okay? There are two basic kinds of prokaryotes, bacteria and archaea. So bacteria, one of the things that they're really known for is having a cell wall that contains peptoglycans. Also, bacteria is what usually causes disease. Not all bacteria cause disease, but some do. They're also, you would think that, okay, well, if these are harmful or causing diseases, why would we want them? But they're also very beneficial. If it wasn't for bacteria, we wouldn't have decomposition. We wouldn't, they're the recyclers of the natural world. That's how we can, because matter can either be created or destroyed, only changed. So if you use it up and you can't recycle it, then all of a sudden it's stuck in this one form. But having bacteria that can recycle everything, you get a chance to get your free carbon, your nitrogen, your sulfur, all of those atoms that you need to survive as an organism. Archaea. Archaea are kind of fun. They are usually known as extremophiles. So if you ever hear about a single-celled organism that's living in on a lake with high salts, on thermal vents, anywhere where you're going where? They will live with high salt, high pH, low oxygen, high temperature. They're called extremophiles because from the coldest places to the hottest places, you can find archaea usually growing. So they're kind of an interesting little one. And currently, to date, we have not found an archaea that actually causes disease. But, oxygen-depleted swamps, find archaea. Eukaryotic structures. So these are fungi. And so we have penicillin on the left. You have this nice hyphae growing, which is the body of the fungus. And then you have the spores, which is how this fungus reproduces. It creates spores. Budding yeast. Saccharomyces cerevisiae is a common example of budding yeast. And they simply bud. That's how they usually reproduce. So they can either bud and reproduce asexually, or if you starve them, they kind of go, oh shit, I should divide because things aren't good, and they'll form tetrads, and they'll make spores themselves, and then they'll go through and they'll do you can mate them. Fungi are kind of cool because they are eukaryotic. Eukaryotes, so the two big classes are prokaryote and eukaryote. Prokaryote, no nucleus. Eukaryote has a nucleus, has a membrane-bound nucleus. One of the big differences. So eukaryotic, they obtain their food from other organs. So this is different than, say, a plant. Plant is able to make its own food using photosynthesis. They also contain cell walls. Animal cells, so us, we don't have cell walls. Plants have cell walls. And fungi have cell walls. But a lot of other eukaryotes, animal eukaryotes, don't have cell walls. So if we're looking at molds, or yeast and stuff, so molds, they're multicellular, they grow long filamentous forms. If you ever have left bread out too long, you see that nice white fuzzy mold, probably penicillin growing. Budding yeast, Saccharomyces cerevisiae, is a common, lovely thing. It's a model organism. It is what makes your bread rise. It's what ferments beer. Not all yeast are pretty and fun and happy. You can have, they have Candida albicans, is apparently the yeast that is most known for causing yeast infections. And then they also have Cryptococcus neoformans, is another form of yeast, which causes cryptococcal meningitis. So you get past the blood-brain barrier in your head and cause meningitis. So not all yeast are pathogenic, but some can be. So, and then if you were going to look at this underneath the microscope side This is more of what you would be seeing because you stain it, have all nice sorts of pretty colors and resin. But you can see the hyphae, you can see the spores very clearly. Protozoa. So our first animals. They're single-celled eukaryotes. They're similar to animals as far as nutrient needs and cellular structure for the most part. Some of them can do photosynthesis, not all. They're free-living. Some actually need animal hosts, a lot of them don't. Most of the time, they will reproduce asexually. Sometimes they will reproduce sexually. And a lot of them are capable of locomotion. So it's kind of cool. You either have pseudopodia, which is where, so pseudopodia is false foot, so they actually will stretch out and kind of anchor on and then pull the rest of the body to it. And so this way they can keep kind of moving themselves along towards maybe a food source or away from something. And so pseudopodia, they do the anchoring movement. You can have flagellum, which is this whip-like structure, which we will talk about a lot more when we get to chapter 3. And so this whip-like structure depends on which organism we're in. Bacteria can swivel around and help it speed through. In eukaryotes, a lot of times it does more of a pulling motion, and it pulls the eukaryote through the media. And cilia. A lot of our animal cells, we have cilia. That's what lines our mucosal membranes. Just a bunch of these little tiny hairs, and they beat rhythmically. This is where, when you cough, you're kind of cleansing that mucus layer. You're getting rid of the dust that your cilia is kind of trying to push along, going, get this out of here, make this go away. And so that's what your cilia is doing. Questions? Okay. Algae. Algae's fun. They can be unicellular or multicellular. Algae is photosynthetic. Nice, pretty green. Usually is dead giveaway. Photosynthetic. They're very simple reproductive structures, and they get characterized based on pigment, how they store things, composition of their cell wall. Large algae are known as kelp or seaweed. So, we've all seen those large kelps, especially if you've gone to Monterey Bay Aquarium, you see their entire kelp forests. They're algae. That's just a multicellular form of it. Algae is incredibly useful scientifically, and because there are some of the chemicals in algae that they're gelatinous cell walls that actually get taken out and used as thickening agents in food, in cosmetics, agar, which will be pouring plates and stuff. So algae's cool like that. Also, single cellular algae, the little tiny unicellular algae, most abundant in the planet because they grow in the ocean, and they are the massive food source. That is the bottom of our food chain for pretty much everything. Everything eats that, and then we eat it. So it's a major food for small and aquatic marine animals. Also, because there are so many of them, they produce most of the oxygen. So a lot of our oxygen is produced in the oceans by these little single-celled organisms. These guys here, the diatoms. Diatoms are interesting with the effect. Anybody ever heard of diatomaceous earth gardening? Okay, some. Okay, so diatoms, they have these little silica glass wall structures, and this gives them a little bit of grit. And so diatomaceous earth is made of fossilized diatoms, and they're also used as like a polishing component. Since they're just small little glass pieces, you can have them, they have them in toothpaste. That little bit of grit you have in toothpaste. They're absorbent in liquids. They have them as stabilizing components in dynamite. So there's a lot of little uses for these diatoms, which are just these little microscopic, come on, we're talking 10 micrometers, little microscopic silicon cell walls. parasitic worms. Just a kind of quick little view. Since they are multicellular, they can be bigger than our red blood cells. Remember thinking how big our cell walls are and everything. One of the things I found kind of interesting with parasitic worms is that they can range from microscopic to 7 meters, which is approximately 23 feet. So that was a little shocking and like, okay, remind me not to eat those. Viruses. Viruses are interesting because when you start talking about what is alive, viruses have a lot of those qualities, but yet they don't do it on their own. So technically, they're not alive. Now, if you remember eukaryotic cell, bacteria cell, viruses are even smaller than the bacterial cells. They're tiny since they're infecting them. So they're much smaller than the smallest prokaryote. They're not even visible with a light microscope, so it's nothing we're going to look at in here. I mean, we can't look at them under a microscope, but they're just little tiny things. They're acellular. They aren't composed of a cell. They're obligatory parasites. They cannot replicate and survive on their own. I mean, they can survive outside of their host, but that's only to really infect. They can't reproduce without a host. And they have a very small amount of DNA. One of the cool things that I was reading recently was because viruses are so small, they've actually come up with bee venom and being able to attach bee venom, and they can block it so that only the HIV particles can touch that bee venom, but the cells can't, and so it's a way of specifically targeting HIV within a person and using that size as an advantage. So it's like, oh. Okay, so that was a general of here's the major life systems we're going to be talking about in class. So when we're starting this, one of the first questions scientists were trying to answer is life spontaneous, or does it have to come from some other life form? You know, and if you think about it, if you were going out and you see this dry creek bed, and then all of a sudden water comes in and it's full of life, you kind of think that life came from mud. It must be spontaneous, you know. So some of the things that we now realize is silly, you don't always realize that until you've had time. So there was the difference between biogenesis and spontaneous generation. So biogenesis, life arises from pre-existing life versus spontaneous generation. So A, biogenesis. You see this A, usually means not or without. That's one other thing. Vocab. Anytime you do science, vocab is huge. Huge part of it. I will make sure that there's vocab in some way, shape, or form on every single test. Because when I started in the sciences, they're like, learning science, you will learn more vocabulary terms than if you were to learn a foreign language. Also, I realize I use jargon. I have been dealing with science for years and years and years. I don't even realize it sometimes. If I say something you don't understand, raise your hand and ask. I can explain it. But I don't realize when I'm using jargon. Okay? Sound good? So, we're looking at the difference between life arising from pre-existing life and life arising from non-life. And so there was a couple of experiments that were done in the beginning. So, new ham, boiled broth, they sealed these containers, and they turned cloudy. Ceballos, he boiled broth infusions, sealed the flask, and the clear broth remained clear. And so, kind of get into, okay, so why were his cloudy, why were his clear, what happened, what does this prove? So one of the thoughts is that, well, he failed to heat his broths properly. He didn't kill everything. That's why they turned cloudy. Or he didn't seal it tightly enough. So that's why it was cloudy. And then, the idea is that, okay, maybe microorganisms can exist in air and contaminate experiments. And so one of the other thoughts was, what about the other guy? He sealed his flask. Well, maybe he didn't, you need to have air. He didn't let his organisms have air so they couldn't grow, which is why it stayed clear. And so, that's why not enough air for the organisms to survive. Or maybe he just boiled them for too long. And so nothing could survive, and nothing could even live in that anymore. And so he was destroying the life force. And so these were all kind of, there was too many problems and holes with those experiments. And so this is where Pasteur came along with his swan-necked flasks and solved or settled the entire debate. Okay. So, Louis Pasteur, he created these swan-neck flasks with broth in them. Pour broth in, attach, heat it up, you can boil it. So, added heat, boiled his broth, made it sterile. Great. But it has that nice swan neck, steam can escape, you can get airflow in and out. Air is not stopped because there is no stopper in this. But if you leave it for months and months and months, you'll eventually get some dust building up, but your broth will stay clear. And then it's like, so look, there is no spontaneous generation. I didn't boil, and then to prove that he didn't overboil this so that it's not capable of supporting life, just tilt it and touch that dust and bring it back up. Or another one was, broke off the neck, and all of a sudden it was cloudy the next day. So either way, if you touch the dust, you break off the neck, you're going to start getting it cloudy the next day. So this showed you have the air, microbes could have grown in here, because they obviously did, but it's really all the stuff in this dust particle that is being carried in with it. Air can get in, the microbes couldn't get in through that swan neck to contaminate the solution. Does that make sense, that train of thought? So this leads us to scientific method, which is very important. It led to, there was a lot of debate about this. And so you have observations. Your observations lead you to questions. Your questions lead you to a hypothesis, which you're then going to try and do an experiment to test, and you either accept or reject or modify your hypothesis based on what your results say. So, try to figure out if is microbes causing this? Yes, no. Anytime you set up an experiment, a good experiment has a yes or a no answer. Did a microbe grow? Yes or no. It's a good yes or no answer. Okay, so breaking down some of them, hypothesis is proposed explanation for a set of observations. An experiment is a test. You always want to use a control observation so that you can try to refute a hypothesis. If you don't have a control, then your experiment's pretty much useless. And then theory. If you have a hypothesis, you do something, proves your hypothesis still seems to be accurate, and over time, if a lot of people have also proven that what you've done, they can't debunk it, then it becomes a theory. So theories are just long-standing hypotheses. They're repeatedly verified, and they can explain a wide range of things. Theories are not always correct, but generally they are, just because it's something that's been known for a long period of time. But science is constantly changing and growing as we learn new things. Okay, so kind of an example of how you would go through the scientific method. So the question was, fermentation, food production. So how do we form sugar? How do we form alcohol? How do we form lactic acid? Okay, so this was incredibly important to the vintners because wine spoiling was ruining their profits. And this is the time when they're kind of thinking, okay, well, we think air causes fermentation of our grapes. And you're kind of going, okay, so how are we, so they didn't know. If you remember the old days of winemaking, you actually have natural yeast on the grapes, which is what fermented the wine. They didn't know that. You crush up your grapes and you get wine. So since it was important to them, they wanted to know how do we stop this from spoiling, how do we make fermentation happen, and so they were the ones who really pushed forward the funding for this. So, first hypothesis, spontaneous fermentation occurs. So, heat up a flask, seal it, wait and see. Nothing happens. So, since it remains free of microbes, you reject the first hypothesis. Obviously, spontaneous fermentation does not occur. Hypotheses do not need to be correct. Most of the time they aren't. They are your first, you go through, you have a question, you have an observation, you have a question, you make a basic hypothesis. Hypothesis 2. Air ferments grape juice. So, you have those swan neck flasks again, heat them up, let air get to them. No fermentation. The juice remains free of microbes. So we reject hypothesis 2. So then go, okay, bacteria ferment grape juice into alcohol, because that's what we really want. We want to know what is making our alcohol. So you heat them up, you inoculate it with bacteria. Bacteria produce acids. You're like, okay, so they did something, but now they want. So in this case, you can modify your hypothesis. Bacteria ferment grape juice into acids, like vinegar, not into alcohol. Let's try it again. Maybe yeast ferment grape juice into alcohol. Inoculate your juice, your grape juice, yeast. Yeast produce or reproduce, the alcohol is produced, and then at the end you get wine. You're like, okay, salt, you can accept this hypothesis. Yeast ferment grape juice into alcohol. So does that make sense, kind of progression of how you would do? Now, what would be the control in this group of experiments? What is a control? Constant. First one. Spontaneous fermentation. Because if you're spontaneously getting stuff to grow there, it doesn't matter what you're adding. Something else is happening. So, if you get growth in one, then all the growth you get and everything else, what does that mean? Something's wrong there. So you always want to have checks throughout your entire system. If you was getting growth in one, then maybe things weren't being sealed right, wasn't heating it up enough, wasn't sterilizing something. But you couldn't accept anything else. Yeah.
Could a control also be thought of as an identical experiment without the variable being tested?